I'd like to express my sincere thanks to University of Oslo, AstraZeneca, and Novo Medium for having this great opportunity. So, as you heard in the introduction, I started my career as a surgeon long time ago, almost 30 years ago. And I started my presentation with a picture of a person, of a man who I respect the most. Actually, because of him, I became a doctor, surgeon. This is the person. Do you know him? Have you seen him in textbooks? I don't think so, because this is my father. So I think he is very handsome. I like his hair, actually. I'm envious. So my father was not a doctor or a scientist. He was an engineer. He had a small factory. And when I was a junior high school student, he got a small injury while he was working on his own factory, which resulted in blood transfusion. So his injury itself was okay. But because of this blood transfusion, after that, he suffered from hepatitis. At that time, he was diagnosed as non-A, non-B hepatitis. So we did not know the cause of his illness. Because we did not know the cause of his illness, of course, there was no cure for his illness, non-A, non-B hepatitis. So he became worse and worse. He suffered from liver cirrhosis. Then he started saying me, Shinya, you should become a doctor. You don't have to take over my own business, but you should become a doctor. I don't know why he said so, or because I was his own son, so I was supposed to succeed his business, but he told me not to do that. Instead, he told me to be a doctor. So I listened to him and became a doctor. In 1987. At that time, he was very sick. He must have been in pain. But when I gave him some small medical procedure, like some transfusion, he seemed to be very happy, even smiling by receiving medical procedure from his own son. So I thought he must be very proud of his own son becoming a doctor. Unfortunately, however, he passed away next year. So he passed away in 1988 when I was 26 years old. So I still needed him a lot. So that, his passing was very shocking to me. I felt powerless, useless. I became a doctor, but I couldn't help my own father. That was, I believe, one of the main reasons why I decided to change my career from a surgeon to a scientist, because I believe it is a medical science who can help those patients, like my father, who are suffering from intractable diseases. So let me talk a little bit about my father's illness. 1 year after my father passed away, this virus was identified in US, hepatitis C virus. It was identified in 1988. 1987. I'm sorry, 1989, I believe. Still in jet lag, I'm sorry. Because now that this virus, hepatitis C virus, was identified, many researchers jumped into researches trying to find cures for hepatitis C. And thanks to those many researchers, very recently, we have a cure. We now have this, sorry, this is in Japanese, but this is Harmony. This is a very nice medicine for hepatitis C. Just one tablet each day can cure almost 100% patients suffering from hepatitis C virus. One tablet for 3 months. That's all you need to do. So it's like a miracle. So this is what we medical scientists want to achieve. We want to overcome diseases by doing science. This is what we want to do. And this hepatitis C, the history of hepatitis C, is a good example of a success of medical science, because now we have overcome hepatitis C thanks to basic medical science. So this is very good. At the same time, the history of hepatitis C clearly shows two problems, two hurdles we medical scientists are facing, two issues. The first one issue was that the virus was identified in 1989, and the cure was developed in 2014. So it took 25 years. It took too long. Well, science did overcome, but it just took too long. This is the first hurdle we are facing. We have another hurdle. We have another problem in medical science. How much do you think this one tablet cost? This is a just small tablet, very, very small. It costs 500 US dollars. I believe it's 3,800 krona in this country. One tablet. So you need to multiply 90. So it's too expensive. So these are the two issues we are facing. It takes too long, and it's too expensive. So overcoming these two problems is as important as overcoming diseases itself. Let me go back to my own science. I was not involved at all in hepatitis C research. I got my PhD in Japan, and I did my postdoc in San Francisco, where I met ES cells, embryonic stem cells. Since then, I have been working on embryonic stem cells, but I realized a big ethical issue about human embryonic stem cells. That is, we need to use human embryos to generate human ES cells. So we worked hard to overcome that ethical issue about the usage of human embryos. Then, in 2006, we were able to publish this paper. From mouse skin fibroblasts, we became able to convert skin fibroblasts into stem cells that are nearly indistinguishable from embryonic stem cells. We designated this new type of stem cells iPS cells, for induced pluripotent stem cells. So the procedure was very simple. All we need is a combination of 4 transcription factors that are listed here: OCT34, SOX2, KLL4, CMYC. We now know CMYC is indispensable. So basically, 3 factors can convert skin cells back into iPS cells, stem cells. In the following year, 2007, we were able to recapitulate same procedure in human adult skin cells. So by having the same combination, we can convert human skin cells back into stem cells, iPS cells. So that means we can make your own iPS cells from each of you. The procedure is actually surprisingly simple. We had a hard time to believe in the beginning. So, but we repeated the same experiment again and again, and to our surprise, it is very reproducible. So in the beginning, we used skin fibroblasts to generate iPS cells. But now we can make iPS cells from many types of somatic cells. The most frequently used cells as an origin of iPS cells is this cell, blood. More precisely, blood lymphocytes. So all you need is just one vial, like 10 ml or 20 ml of blood, peripheral blood sample. From this, by adding that combination of 3 or 4 factors, we can convert lymphocytes into iPS cells. As you know, lymphocytes cannot proliferate to a large amount. We can expand a little bit, but there's a limitation. And also, lymphocytes, no matter how long we wait, they are lymphocytes. They cannot become a heart cell or a brain cell. But by adding those transcription factors, we can convert blood cells into iPS cells. So this is a colony of iPS cells. I believe we have approximately 500 iPS cells in this one tiny colony. Each iPS cell is like 10 micrometer. So one of the smallest cells, one of the smallest cells. But they're very small in size, but their potential is enormous. Once again, they have 2 very important properties. First of all, we can expand iPS cells as much as we want. As you may know, we culture iPS cells or other types of cells in this kind of plastic dish, Petri dish, like 10 centimeter. From a very small amount of blood cells, we can have hundreds or even thousands of Petri dishes containing iPS cells, your own iPS cells. And after expanding to a large amount, by treating these iPS cells with some cytokines, growth hormones, we can convert iPS cells into many types of cells. For example, we can make this kind of beating heart cells in hundreds of Petri dishes from iPS cells. They're beating, they synchronize. So we can see them beating even without microscope. Considering these cells used to be skin or blood cells, even now I feel a bit strange whenever I see them beating. Oh, they have stopped, but. So, once again, our vision is to overcome diseases by doing science. So by using iPS cells, we want to overcome intractable diseases. There are two major ways to do that. One is regenerative medicine, also known as cell therapy. The other application is drug development. So for example, my father passed away from liver failure. We could help patients suffering from end-stage liver failure by transplanting livers. But in countries like Japan, organ transplantation is next to impossible, because in Japan, brain death is not accepted. So like liver or heart transplantation is very, very, very limited in Japan. But by applying this technology, we can make iPS cells from my father's blood cells. We can expand my father's iPS cells as much as we want. Then we can convert, at least in theory, my father's iPS cells into his liver cells. And then instead of liver organ, we can transplant healthy liver cells back into my father, back into patients. So that's how we do in regenerative medicine. Also, by using this technology, we can prepare a large amount of human liver cells. Otherwise, it is very next to impossible to obtain a large amount of human liver cells. I believe that was one reason why it took 25 years for a drug company to develop Harmony. With this technology, now we can have researchers at pharmaceutical companies like AstraZeneca have a large amount of human liver cells, heart cells, or brain cells. So that should facilitate drug development. So with this technology, I believe we could have shortened that 25 years. I don't know how much. It could be, 20 years or maybe 15 years. But that's how iPS cells can contribute to drug development. So let me give you a few more examples about each of these two major medical applications. Let me begin with regenerative medicine. It's been 10 years since we reported the first generation of human iPS cells. To my surprise, one group in Japan has already started a clinical trial using human iPS cells. That is Dr. Masayo Takahashi. She is an ophthalmologist and also a very famous famous neuroscientist. So she has been working on age-related macular degeneration, in which a layer of retinal cells, known as retinal pigmental epithelial cells, becomes degenerated because of aging. Because of that, patients are losing vision. Her team, Dr. Masayo Takahashi's team, generated iPS cells from patients' own skin cells. And then they converted iPS cells into a sheet of retinal pigmental epithelial cells which is shown in this Petri dish. And they transplanted this small piece of epithelial or retinal pigmental epithelial cells back. They transplanted this small sheet back into patient's eye. They performed the first clinical trial, first surgery, almost 3 years ago in 2014. And the patient is doing very well. Before the surgery, her vision was getting worse and worse every month. But after this surgery, it stopped. So her vision is now very stable. Whereas the opposite side, to which she did not get this surgery, it is almost blinded by now. So I would say this first transplantation went very well, was very successful. However, she stopped after this first patient. There are two major reasons. One reason was a legal reason. In 2015, our country, Japan, established a new law regarding regenerative medicine to ensure the safeness of regenerative medicine. Because of that new law, Dr. Masao Takahashi had to start from the beginning, all paperwork. So that was one reason she had to postpone this clinical trial. But there's one more scientific reason. We helped this clinical trial in many aspects, and we found this kind of cell therapy using patients' own iPS cells was too expensive. It took almost $1 million for just one patient. So we thought it was too expensive. And it took almost a year to do all the processes required to generate iPS cells from the patients, to do quality control of iPS cells, to differentiate iPS cells into retinal cells, and to do final quality checks of retinal pigmental epithelial cells prior to transplantation. In total, it took almost a year. Within that year, patient's condition can change. For other patients, like patients suffering from heart failure or liver failure, many of them cannot wait for 1 year. They may die. So once again, the cost and also time required for autologous transplantation was the biggest hurdle we learned from this first clinical trial. In order to overcome those two practical hurdles, we are now working on this project. iPS cell stock for regenerative medicine. So in this project, instead of making iPS cells from patients' own blood or skin cells, we are making iPS cells from healthy donors. So it's allografts. It's not autologous transplantation. Since it's not autologous transplantation, we can save time and money, of course. But we need to overcome immune rejection because we don't use patients' own iPS cells. In order to minimize immune rejection, we need to match immunological type between donor and recipient. More precisely, we need to match HLA haplotype. However, HLA haplotype is very diverse. There are more than 10,000 haplotypes. So none of you in this audience has the identical HLA haplotype, unless we have an identical twin in this room. So that means we need to prepare thousands of iPS cell stocks. It's not practical. Instead, we are making iPS cells from so-called super donors. Super donors are HLA homozygous donors. I won't go in detail, but those super donors are super because when we transplant their cells, like iPS cell-derived retinal cells, iPS cell-derived brain cells into recipients, we can minimize, we can expect very small amount of immune rejection. So that's why we call them super donors. So we are now making iPS cells from these super donors. But super donors are very rare. It's usually 1 out of like 500 people. So we may have 1 or 2 super donors in this audience. But in order to identify those 1 or 2 super donors from this audience, we have to determine HLA haplotypes of all of you. It's a lot of work. But very luckily, we are now getting help from Japan Red Cross, who has been working on programs like platelet transfusion or bone marrow transplantation, and also cord blood banks. So that means they have a huge database of Japanese HLA haplotypes already, close to 1 million Japanese people. So now that we can access to their huge HLA database, we can easily identify these super donors. So once we identify these super donors, we get informed consent from those potential donors. And once we get consent, we get blood samples. Then we transfer those blood samples in our CPC, cell processing facility in our research institute in Kyoto, where we are making clinical grade, GMP grade iPS cells. We have enough time to perform a rigorous quality check, and we only ship high quality iPS cell stocks to other researchers in academia as well as industries. So far, we have generated iPS cells, clinical grade iPS cells from 2 super donors. I would say they are super donors because just 2 donors, iPS cell lines from just 2 donors can cover up to 30% of all the Japanese population. So that means just 2 donors, they could help up to 30 million Japanese people. So they are really super. So once again, this is the process of autologous iPS cell transplantation. Once again, it took too long and it is too expensive. But by applying our iPS cell stock project, we can skip this initial portion. So that means we can lower the cost and we can save time tremendously. Once again, we have already shipped iPS cell stocks from 2 super donors, and to our delight, the same doctor, Dr. Masao Takahashi, has already started clinical trial using our iPS cell stocks. She performed operation this March 28th, this year. So because this is allografts, this utilizes iPS cell stocks. That means we can transplant cells to many patients simultaneously. In autologous transplantation, we can do only one by one, so it would take very long. But because this is allografts, we can transplant to multiple patients. She started this first patient in March, but she has performed many more patients already. She will publish it when it's ready, but I would say she's doing multiple patients by now. So in addition to retinal disease, many projects are going on in Japan and also in many other countries using our iPSO stocks. For example, Parkinson's disease, like corneal disease, heart failure, spinal cord injury, blood transfusion, cancer, and also arthritis. Blood transfusion, now we can get enough blood from donors, healthy young donors. But as you know, Japan is a graying society. Japan is probably the fastest in terms of aging, and also we are having less and less younger generation. So in countries like Japan, we are facing a huge problem. In 5 years, we will short on blood donors. So millions of people would die because of the shortage of blood transfusion. It's a huge, huge problem. So this application, we are now making platelets as well as red blood cells from iPS cells. This should help that kind of shortage of blood donors. And also we can make T lymphocytes attacking cancer cells from iPS cells. That would facilitate cancer immunotherapy, which is a very trendy therapy in cancer. So let me move on to the second application of iPS cells. It's about drug development. So as I mentioned previously, we can prepare a large amount of human liver cells, heart cells, and brain cells. Those cells can be utilized in any types of drug development. For example, like Alzheimer's diseases. But in today's presentation, I would like to focus on another type of diseases, which is known as like rare diseases. One example is this, ALS, amyotrophic lateral sclerosis. This is a form of motor neuron diseases. So this is a typical symptoms of patient. Patients progressively lose their muscle movement, beginning with their fingers, then move to their speech. In the end, they cannot breathe without help of respirator. Because motor neurons progressively die, they cannot, so the brain message cannot be transmitted to peripheral muscle. So muscle itself is not sick. It's motor neuron disease. But because of that, their muscle become very atrophic. In US, this disease is well known as Lou Gehrig disease. Lou Gehrig was a very famous and popular baseball player, long time ago, 70 years ago. But in one season, he suddenly became unable to hit well. So everybody, including himself, thought he was in slump. But in reality, he suffered from this ALS. And he had to retire because of ALS, and he passed away just a few years later. So this became a very popular movie in US, so that's why this disease is well known as Lou Gehrig disease in US. It's been more than 100 years since we got to know about ALS. Many scientists have been fighting with ALS in order to overcome this terrible disease. But it has been unsuccessful. So this is kind of a symbol of a failure of medical science. We have a mouse model of ALS, and many scientists and many companies have developed very effective drugs for mouse patients. But unfortunately, the same drug did not work at all on human patients. So in the case of ALS, we need to work on human motor neurons, not mouse models. But as you can imagine, it is next to impossible to obtain human motor neurons. Because if you get motor neurons from donors, the donor will lose his ability to move his muscle or her muscle. So we cannot do that. We may be able to obtain motor neurons during autopsy, but motor neurons do not proliferate at all. They are terminally differentiated. So we cannot do a large experiment on such motor neurons. That's probably the major reason why it's been unsuccessful in fighting with ALS. But now we can use iPS cells. Many researchers in the world are now making iPS cells from ALS patients, and also they are successfully making motor neurons from patients' iPS cells, including Haruhisa Inoue, one of my colleagues. He is a neurologist who have been working on ALS patients. So even before we reported our mouse iPS cell generation, he saw many ALS patients. Many of them died, but he took skin fibroblasts from those patients, hoping in future those skin fibroblasts would help. But at that time, there was no way to utilize those skin fibroblasts because they are not motor neurons. But as soon as we published our mouse paper, he came to me, and we started long-lasting collaboration. So as soon as we successfully generated human iPS cells, he utilized his patient's skin fibroblasts in our protocol. So he was one of the first to generate ALS patient iPS cells. So in this disease, usually patients are fine until their 50s or 60s. So we thought probably it would take very long to recapitulate disease by using iPS cell-derived motor neurons. If we culture them for 50 years in Petri dish, we may be able to recapitulate. We may be able to shorten by giving motor neurons some kind of stress. But in reality, our prediction was wrong in a good way. As soon as Dr. Inoue generated motor neurons from patients' iPS cells, they start dying automatically without any stimulation. Whereas motor neurons from healthy iPS cells, motor neurons wouldn't die. So that means we can recapitulate motor neuron disease in Petri dish, at least to some extent. And now that we can have patients' motor neurons in hundreds of Petri dishes, we can test different drug in each of hundreds of Petri dishes. That's exactly what Dr. Inoue did, and he found one existing drug, bosutinib, which is a clinically available drug for leukemia, is actually effective on preventing motor neuron death from ALS patients. So this approach is now known as drug repurposing or drug repositioning. So this is very powerful because we can shorten the process and the money of drug development because they are already being used with patients. So we are hoping we can utilize this finding with iPS cells so that we can generate effective drugs for ALS patients as quickly as possible. Bosutinib itself is a cancer drug, so it has a strong side effects. So at the moment, we are not sure whether we could or we should use this drug in ALS patient. We have been talking to many people, like people from the government. We have to be very careful, but at least we can utilize this finding to search for new drugs. Let me give you one more example, which is a very rare disease, FOP, fibrodysplasia ossificans progressiva, FOP. We only have less than 100 patients in Japan. Worldwide, there should be less than 1,000 patients. I don't know how many patients in this country, but probably less than 50. So in this patient, their muscles, tendons, ligaments become bones progressively. So in the beginning, when patients are very small, they're okay. But when they become like high school students, they have bone everywhere. So they cannot move. And in the end, they cannot breathe. So it's a very also terrible disease for patients and also for family members. It's a very rare disease, but we met one patient in Japan 7 years ago. This is the patient. When I met him for the first time 7 years ago, he was still he was very small, like 10 years old, and he was fine at the time. But 7 years later, he suffered from multiple bone formation. He may look okay, but he's very skinny, actually, because he has many bones in her face, so he cannot open her mouth. That means he cannot eat. So that's why he's very tiny. He often comes to our institute, and whenever he's with us, he asked us to take pictures. And he asked us to do this posture. In the beginning, I had no idea what this means. What do you think this means? Do you think this means, Dr. Yamanaka, please be #1 runner? Of course not. So this means he wants us to develop an effective drug as early as possible, even 1 day earlier. So it's a very strong will of him and also his mother. He precisely knows most likely we cannot make it for him. But he wants us to do it for patients after him. So, but Professor Tokuchira, a good friend of mine, he generated iPS cells from this patient and also from many other patients, trying to understand why on Earth they generate ectopic bones in their muscles. And now they have clues. So they, to some extent, they were able to recapturate ectopic bone, excessive bone formation by using patients' iPS cells. So for example, from patients' iPS cells, from mesenchymal stem cells derived from iPS cells, they can make cartilage. And patients' mesenchymal stem cells tend to generate more cartilage than normal iPS cells. Of course, cartilage is one step earlier than ectopic bone formation. And they also found that activin A is involved in this process. By having this result in mind, they performed drug screening, and they found that this existing drug, rapamycin, is very effective in preventing ectopic bone formation from patients' iPS cells. They confirmed this finding with iPS cells in vivo using mouse model. So by transplanting FOP mesenchymal stem cells derived from patients' iPS cells into mouse muscle, and by adding activin A, they observed this ectopic bone formation in mouse muscle. But by treating the same mouse with rapamycin, they did not see such ectopic bone formation. So rapamycin is being used for patients already, including small children. So that means we can translate this finding with iPS cells into patients very quickly. As a matter of fact, his application has been approved by the Japanese government. This is September 6th today. So as of tomorrow, September 7th, they will start clinical trial for patients suffering from FOP, including that patient. So that means we may be able to make it for that patient. At the moment, it's also successful in Petri dish with iPS cells and also just in mice. So we need to wait for the result of this clinical trial. But I believe it's very promising. So let me finish my talk by introducing this very unique collaboration with a pharmaceutical company. Takeda is the largest pharmaceutical company in Japan. Of course, I know it's smaller than AstraZeneca, but it's largest in Japan. So we have started this collaboration 1.5 years ago. This is very unique. You know, collaboration between academia and pharmaceutical companies are very common. I believe you have many, many collaborations. But this is very unique because the direction is opposite from conventional collaborations. In conventional collaborations, researchers of pharmaceutical companies, they come to universities to make some collaboration, to make some experiments together in academia. But in this collaboration, we go to Takeda's huge research facility in Tokyo area in Shonan. It takes us like 2 hours from Kyoto to Shonan, but it's worth doing. 7 professors from our institute, they spend like 20% of their efforts every week in Takeda's research institute. They make a team with Takeda's employees. So because we go to Takeda, inside pharmaceutical company, everything is available. Not only their research, their chemical libraries, huge libraries, we can access to everything they have, including their experiences in drug development, many other experts in many different areas. So we found this very, very useful. So I found I hope by doing this kind of new type of collaboration between academia and pharmaceutical company, we can promote translation of academia-driven researches like iPS cells into patients. So I have been leading this so-called T-CIDA project, Takeda CIDA project. My counterpart of Takeda is this scientist, Dr. Seigo Izumo. So 30 years ago, I was looking for a postdoc position in the States. And the number one position, lab, to me, was in Harvard University. That lab was run by Dr. Izumo, so same person. 30 years ago, he didn't take me. I was so sad. But 30 years later, we met again, and we worked together, so we are very happy. Well, who knows? So, thank you so much again. We have more than 600 people in Kyoto. We have more than 100 people in this T-CIDA joint program. And I also have a small group in San Francisco. I will be in San Francisco next week. So all of them are very important colleagues of mine. We have a common vision. We want to overcome diseases by doing science. And I believe it's the same to you. And finally, I would like to express my sincere thanks to Dr. John Gurdon. So thank you very much again. Thank you for your attention. Thank you.
Thank you very much. Thank you so much for sharing that story with us. I think it's very fascinating. These basic discoveries at the beginning of this century is already having such tremendous impact in medicine. So Professor Yamanaka has agreed to take a few questions. There should be 2 microphones, one on either side. So if you have a question, raise your hand and wait until you have the microphone before you ask the question. You only speak into the microphone because this is also being recorded. So we can take questions.
Thank you. Dr. Yamanaka. It was a really inspirational talk. I want to ask you how you see this going in the future. Do you see, for example, combining iPS technology with genome editing, for example, to fine-tune the properties of your iPS cells?
Yes, that's very important. Combination with this technology and genome editing technology is very promising, and that's exactly what we have been working on. Yes. That's one reason why I go to San Francisco every month, because Bay Area is one of the hottest area for genome editing.
There was another one in the front here.
Yeah, thank you. Arne Sørås, I'm working in epigenetics, and how much is known about epigenetics, especially about the reprogramming process? Is it known how it happens, or is it very much unknown?
That's also extremely important. So each cell fate has unique epigenomic status, and we and others have found that during iPS cell generation, most of epigenomic status is reverted, or I would say erased back into the embryonic state. But it's not 100% complete. So we do see some abnormal or like DNA methylation. So we are now studying the impact of such epigenetic abnormalities in iPS cells. So far, we believe it doesn't affect too much, but we still need to do a lot more research. So it's extremely important.
There's one.
Is any structure analysis for that it's done to show the differences between what makes stem cells different than normal cells?
Yes. So, for example, stem cells, especially pluripotent stem cells, can proliferate for almost infinitely. So we and others have shown that, in a sense, ES cells and also iPS cells are similar to cancer cells, in that they have a very high telomere activity. But as soon as they are terminally differentiated, they lose their telomere activity. So, and many signaling pathways that function in cancer cells are also activated in pluripotent stem cells. So, compared to normal somatic cells, ES and iPS cells are kind of in between normal cells and cancer cells. But it's reversible. Cancer cells are not reversible because they are formed by DNA mutation, whereas ES, iPS cells do not have such mutations, so it's reversible. So that's what we know at this moment.
While we're waiting for that, I'd like to ask another question, because you sort of, in transplantations, immunosuppression is often needed, maybe not in the eye, but in other sites. And so the great thing about using the patient's own cells is you don't need immunosuppression. But now you have sort of switched from that idea to use sort of donors for that. Do you think that this field will move to that sometime in the future they will be using the patient's own cells? Was that going to be more cheap and faster, or yes. Is that something that you're also working on?
Yes, that's a very good point. That's exactly what we want in the future. At the moment, using this kind of iPSC stock, I believe, is the way to go because of its price and time required. But we are doing our best to promote our method to generate iPS cells.
So that's something you also.
Yes, once they become more, much more quicker and cheaper, I think autologous transplantation is the way, should be the way to go.
There was one in the middle there.
Question is was related to the same thing. So for the cell therapies, are there certain conditions or diseases where you think it's absolutely necessary to use the patient's own cells, even today? Or can you use the stock cells for everything?
So because of the development in immunosuppressants, I think we can go with allografts for most, if not all, diseases, patients. But it would be much better if we could use autologous transplantation because we can avoid any immunosuppressants.
Plant cells are much more easy to reprogram and are much more pluripotent than animal cells. Do you have a viewpoint on the reason behind this, the mechanism behind that? And could one learn something in the medical science from the plant science, possibly?
Well, that's a fundamental question. So many scientists have been trying to understand what's going on during iPS cell generation. I would say largely it's still like a black box. For example, we now know that, at least in human, during iPS cell generation reprogramming, endogenous retroviruses play a major role. So as you know, we have more than 3,000 endogenous retroviruses in our genome. Until very recently, we thought those are just junk. But to our surprise, we found many of them activated during iPS cell generation. And we found it is not just coincidence. We found the activation of endogenous retroviruses are essential for iPS cell generation. But we don't know why they are essential. So, and also endogenous retroviruses are not conserved between human and mouse. But even without endogenous retroviruses, we can generate mouse iPS cells. But in human, endogenous retroviruses are essential. So we still don't know the answer. That's what I have been working on in my San Francisco lab for the last 5 years. Basic aspect, basic question, why endogenous retroviruses are important in human. So if you can wait for 5 more years.
We have one from the second row here.
Yeah. Thank you, Professor, for coming to Oslo and sharing your story with us. My question would be on longevity research. So Japan is having a huge amount of population who are actually aged. So since you are in the forefront of this research, do you think that you would be allowed to work, or you would be allowed to conduct clinical trial on helping people to live longer or healthier?
So our sole purpose is to overcome diseases like heart failure or Parkinson's disease or retinal or macular degeneration, so that patients, so that people can live healthier and happier. So we don't have any intention to make our longevity itself longer. But as a result of overcoming diseases, I think average longevity should become longer. I read a very interesting paper earlier this year about hematopoietic stem cells. So all of our red blood cells, lymphocytes are derived from hematopoietic stem cells. And in young people, like you, we believe we have approximately 10,000 hematopoietic stem cells. But that paper studied how many hematopoietic stem cells are there in one very elderly woman who is actually 115 years old. And how many did you think they found? He knows the answer. They found only 2. So without those 2, she cannot survive. So I think that's the limitation of how long we can live. You have 10,000. After 100 years, you have only 2. Unless we perform bone marrow transplantation, you cannot survive.
There was a question on that side there.
Thank you very much for the excellent talk. My field is in the organ transplant, and I'm very curious what you think of using the IPS to building organs. Do you think we should do the maturation of the cells in vitro, or should that be done in vivo to kind of get the cells to become what you want to be? And also, what you think about encapsulation of the cells to control the immunity?
So your question is how to make organs from iPS or other stem cells. Actually, 3 types of researches are going on. I myself are not working on that, but many other scientists are working very hard on 3 different strategies. One approach is to make human, like pig chimera, injecting human iPS cells in pig blasts. Those pigs are genetically engineered so that those pigs themselves cannot make like pancreas or kidneys. So that means they can survive. But by putting human iPS cells into their embryos, those human iPS cells can make kidneys or pancreas in pig. And of course, because they are from human iPS cells, they are human pancreas or human kidney. It's been partially successful. So that kind of research has been conducted in US and also in Japan. There have been ethical controversy about how much we can do that. But the research is ongoing. The second approach is to use 3D printer using various cells from iPS cells or ES cells as ink of making 3D organs. And 10 years ago, I thought it would be like science fiction. But now there are multiple venture companies working on that. So it's also making a significant progress. The third approach is to utilize cells' automatic ability to form 3D structure. So stem cells can make mini brains or mini gut. It's still very small, but I think it's fascinating, actually. they form 3D structure by themselves. I don't know how they are being programmed that way. So it's amazing, but it seems working. So one of those three approaches may work in the future.
We're moving towards the end, but we have 3 more questions. I think we have one there in the middle.
Thank you again for the elegantly updated lecture. I would like to ask you, how would you compare iPS-derived cells from the same patient when the same cell is available? For example, extracted from your talk, mesenchymal stem cells.
So actually I had a hard time to hear, but you asked.
So how would you compare iPS-derived cells to the same cell derived from the same patient? For example, mesenchymal stem cells.
Oh, I see. Yeah, it's very important. In theory, at least in theory, we can make many types of cells from iPS or ES cells. But in reality, it's still very difficult. For example, we can make beating heart cells or dopaminergic neurons from iPS cells. But how similar they are with normal cells that exist in our body depends on each cell type. For example, retinal pigmental epithelial cells, they are very similar to what we have in our body. Whereas beating, also dopaminergic neuron, they are very similar to what we have in our brain. But like heart cells or liver cells, they are still very immature. They are more like fetal heart cells or liver cells, what we can now make from iPS or ES cells.
What about stem cells, mesenchymal stem cells, hematopoietic stem cells?
Hematopoietic stem cells, it's a hot topic. It's a hot topic. The problem about hematopoietic stem cells is that we can take out hematopoietic stem cells from patients, like bone marrow transplantation. But nobody has successfully keep them in culture. So nobody can expand normal hematopoietic stem cells in Petri dish. So we don't know how to culture them. Without that knowledge, it is next to impossible to generate hematopoietic stem cells from like ES or iPS cells. So it's still, the research is still ongoing, but it's very hot topic. Competition is very hot. Don't touch.
Do we have one question from the aisle here?
Yes, my name is Øystein. I made a children's fact book about stem cells, and you are in it. Sorry for not asking. I didn't have your number, but you can have one afterwards. But I'm going on a big tour now to speak to kids all over the country, and I'm wondering if this is like a general of everything going on in stem cells that I could figure out for 10-year-olds. But do you have anything I should tell children now about stem cell research so they'll be like super up to date when they grow up?
Oh, it's probably the most difficult question. Well, all I can say is what you're doing is very important to educate, right? So just keep going, okay. Please.
Then the last question, I think, was,
Thanks for the lecture. When you were talking about the ALS, you said that you were kind of surprised in a good way when you see that the differentiated iPS cells, when you differentiated them into motor neurons, they still suffer from the disease. It's known how this happens. How can the cell remember after differentiating that disease? Thanks.
It's also a fundamental question. So, so any disease is caused by genetic factors and also environmental factor. So probably the second one will affect epigenetic status. And as I answered in the previous question, epigenetic status is erased upon iPS cell generation. So only genetic information is maintained after iPS cell generation. So the fact that we can recapitulate, at least to some extent, disease phenotype in ALS-derived iPS cells, that means those patients should have some genetic background. Like 10% of ALS patients is caused by just one mutation of just one gene. So they are called familial ALS, but it's only 10%. But even from the other 90% of ALS patients, we can recapitulate disease. So that means although they are not familial, they should have some genetic background. So probably it's a combination of multiple genetic alterations, which we don't know yet. But from our result, it is clear that they should have some kind of genetic background. So if we can understand what's going on in their genome, we may be able to identify better strategy to overcome.
Once again, thank you very much for the lecture. It certainly engaged the audience. Thank you for answering all the questions. And there will be a debate in just half an hour in the Science Library for those that are interested in that.
Thank you. So, tusen hjärtligt tack. Thank you.